The neuroscience of jealous emotions. The experience of jealousy involves a complex orchestration of neural activity that spans multiple brain regions and neurotransmitter systems. Modern neuroscience has begun to unveil the intricate biological machinery underlying jealous emotions, revealing how our brains process perceived romantic threats and generate the intense, emotional and behavioral responses that characterize this complex emotion. When we experience jealousy, our brains undergo dramatic changes in activity patterns that reflect the emotion's multifaceted nature. Brain imaging studies using functional magnetic resonance imaging and positron emission tomography have identified several key neural networks that become active during jealous episodes. The prefrontal cortex, particularly the ventromedial and inferior frontal regions, shows increased activation as the brain works to process and evaluate perceived relationship threats. This increased prefrontal activity reflects the intense cognitive effort involved in analyzing potential threats and planning appropriate responses. The basal ganglia, including the dorsal striatum and nucleus accumbens, also show significant activation during jealousy experiences. These regions are typically associated with reward processing and habit formation, but their activation during jealousy appears to reflect the brain's attempt to process the potential loss of a rewarding relationship. The increased activity in these areas may contribute to the obsessive and repetitive thought patterns that often characterize jealous episodes, as the brain repeatedly processes the perceived threat and its implications. One of the most significant findings in the neuroscience of jealousy has been the identification of asymmetric brain activation patterns. Research using transcranial direct current stimulation has revealed that stimulation of the left frontal cortex increases reported feelings of jealousy following rejection experiences, while stimulation of the right frontal cortex does not produce the same effect. This suggests that jealousy processing is lateralized in the brain, with the left hemisphere playing a particularly important role in generating jealous emotions. The limbic system, including the amygdala, hippocampus, and cingulate cortex, shows complex patterns of activation during jealousy experiences. The amygdala, which processes fear and threat detection, becomes highly active when individuals imagine their partners being unfaithful. This activation likely contributes to the intense fear and anxiety that accompany jealous episodes. The hippocampus, involved in memory formation and retrieval, may contribute to the vivid and persistent memories of perceived threats that often plague jealous individuals. The anterior cingulate cortex, which plays crucial roles in emotion processing and pain perception, shows particularly strong activation during jealousy experiences. This region appears to process the emotional pain associated with perceived relationship threats, and its activation may contribute to the intense psychological suffering that jealousy can cause. Some researchers have suggested that jealousy literally hurts because it activates brain regions normally involved in processing physical pain. The insula, a brain region involved in interoception and emotional awareness, also shows increased activation during jealousy experiences. This activation may contribute to the intense bodily sensations that often accompany jealousy: the racing heart, the churning stomach, the tight chest, as the brain monitors the physical manifestations of emotional distress. The insula's role in empathy and theory of mind may also contribute to the vivid mental imagery that jealous individuals often experience when imagining their partners with others. Neurotransmitter systems play crucial roles in mediating jealous emotions. The dopamine system, which is heavily involved in reward processing and motivation, shows altered activity during jealousy experiences. Decreased dopamine signaling may contribute to the depressed mood and lack of pleasure that often accompany jealousy, while increased dopamine activity in certain brain regions may contribute to the obsessive thoughts and behaviors that characterize jealous episodes. The serotonin system, which regulates mood and impulse control, also appears to be involved in jealousy. Low serotonin levels have been associated with increased aggression and impulsivity, which may explain why some individuals become violent when experiencing intense jealousy. Medications that increase serotonin availability, such as selective serotonin reuptake inhibitors, have shown promise in treating pathological jealousy, supporting the role of serotonin in mediating jealous responses. The oxytocin system, often called the love hormone, shows complex patterns of activity during jealousy experiences. While oxytocin typically promotes bonding and trust, research has revealed that it can also increase in-group favoritism and out-group hostility. During jealousy episodes, oxytocin levels may increase as the brain works to strengthen bonds with the primary partner while simultaneously increasing negative feelings toward perceived romantic rivals. Recent research has begun to explore how these neural systems interact to produce the complex experience of jealousy. The default mode network, a set of brain regions that are active during rest and introspection, shows altered patterns of connectivity during jealousy experiences. This may contribute to the rumination and self-focused thinking that often characterize jealous episodes. As individuals become increasingly focused on their own thoughts and feelings rather than external reality. The mirror neuron system, which allows us to understand and empathize with others' experiences, may play a role in the vivid mental imagery that accompanies jealousy. When jealous individuals imagine their partners with others, mirror neuron activation may make these imagined scenarios feel almost real, contributing to the intense emotional responses they generate. This system may also contribute to the social pain of jealousy, as individuals vicariously experience their perceived exclusion from their partner's attention and affection. Gender differences in brain activation patterns during jealousy have been documented in several studies. Men and women show different patterns of neural activation when imagining sexual versus emotional infidelity, with men showing stronger activation in regions associated with physical and sexual processing when imagining sexual infidelity, and women showing stronger activation in regions associated with emotional and social processing when imagining emotional infidelity. These neural differences provide biological support for the gender differences in jealousy observed in behavioral research. The neuroscience of pathological jealousy reveals how normal jealousy systems can become dysregulated. In conditions like Othello syndrome, brain imaging studies have revealed alterations in frontal striatal circuits, particularly involving the ventromedial prefrontal cortex, which is crucial for reality testing and impulse control. These alterations may explain why individuals with pathological jealousy maintain their beliefs in their partners as infidelity despite contradictory evidence. Neuroplasticity research suggests that repeated experiences of jealousy can actually change brain structure and function. Chronic jealousy may lead to structural changes in regions involved in threat detection and emotional processing, making individuals more susceptible to future jealous episodes. This may explain why jealousy can become a habitual response pattern that is difficult to break without intervention. The developing field of social neuroscience is providing new insights into how social context influences the neural processing of jealousy. The presence of social support, the nature of the perceived threat, and the individual's relationship history all influence how the brain processes jealous emotions. These findings suggest that jealousy is not simply an individual psychological phenomenon, but a complex social-emotional process that is shaped by environmental factors. Understanding the neuroscience of jealousy has important implications for treatment approaches. Neuroplasticity research suggests that therapeutic interventions can actually change the brain patterns associated with problematic jealousy. Cognitive behavioral therapy, mindfulness-based interventions, and pharmacological treatments all show promise for modifying the neural circuits involved in jealous responses. The neuroscience research also provides validation for individuals struggling with jealousy, demonstrating that their experiences reflect real biological processes rather than character flaws or moral failings. This understanding can reduce shame and self-blame while encouraging people to seek appropriate treatment for problematic jealousy. As neuroscience technology continues to advance, we can expect even more detailed understanding of the biological basis of jealousy. Real-time brain imaging studies are beginning to reveal the moment-by-moment neural processes that occur during jealous episodes, providing unprecedented insight into this complex emotional experience. This research will likely lead to more targeted and effective treatments for individuals whose lives are negatively impacted by excessive jealousy.